As we learned on the previous slide, there are all kinds of viruses infecting all kinds of different organisms and across all the kingdoms. And they all pretty much infect in the same way. They first trick the cell by binding to receptors on the cell surface, and they have proteins. So, for example, here we're looking at a bacteriophage infecting a bacteria, and it's the tail fibers, the proteins that make up the tail fibers that would be binding to receptors on the bacterial cell membrane. And they trick the cell into letting the genetic material of the virus get inside the cell. In a human virus, it might be glycoproteins that are studded on the membrane envelope of the virus that will trick the cell into bringing in possibly the whole virus. But usually it's the membrane envelope that's going to fuse with the cell, the plasma membrane of the cell that's being infected. And then the capsid will enter. Just the capsule, or the capsid of the virus will enter. An example of the kind of receptors that it might bind to are the receptors that are involved in receptor-mediated endocytosis, if you'll recall. That's something that we learned about in the last unit. In general, once the viral genetic material is inside, and remember it can be either DNA or RNA, it is copied and expressed. But it may be a while before it is expressed. So it depends on the virus. Infection may be lytic or lysogenic. Lytic on the left, lysogenic on the right. In lytic infection, the viral genetic material is expressed right away. In lysogenic infection, the viral DNA becomes part of the host cell genome as a prophage. So here we see that viral DNA has been incorporated into the chromosome of the host cell, and we call that a prophage. It's not bacterial DNA. It's viral DNA that is now part of the bacterial chromosome. The most important difference is that lytic infection results in rapid destruction of the cell, and in lysogenic infection, the prophage may be copied and passed to daughter cells for generations, and that's what they're showing here in the diagram. This cell is reproducing, undergoing binary fission, splitting in two, and the two daughter cells are receiving copies of the prophage in their chromosome. In other words, the prophage is being copied along with the chromosome prior to the splitting of the cells. And they're not showing two cells as they go along in the diagram. They're just showing one of these daughter cells that result from this bacterial reproduction, binary fission. But eventually, the provirus can exit the bacterial chromosome and be expressed. So that can take a long time. So virulent viruses use lytic infection and make you sick quick. A human example would be the common cold. If you catch a cold virus, you're going to be sick within a week or two. In lysogenic infection, temperate viruses use lysogenic infection. So again, it might be a long time and many generations of cells reproducing or undergoing cell division before the viral DNA exits the chromosome of the host and then is expressed. Temperate refers to time, so there can be a long time. HIV is an example of a temperate virus. So the latent period is a term that's used to refer to that period of time before the prophage, or in the case of HIV or a human virus, a provirus, exits the host chromosome and then is expressed. For HIV, that can be 10 years. What triggers the provirus or prophage to exit the host chromosome is some kind of environmental cue or stress. The end result is the same, though. It results in destruction of the host cells. So again, in the case of HIV, the host cell would be T cells, part of the immune system. And it's the destruction of those cells that actually causes the disease, AIDS, acquired immunodeficiency syndrome. Here we see a diagram of how HIV infects a human T cell. And it's more specifically known as a human CD4 helper T cell. And it's named, the CD4 comes from a protein receptor that is on the surface or on the plasma membrane of those types of cells. It's called a helper T cell because it helps the immune system recognize and respond to infection. So these are the cells that are destroyed by HIV that eventually leads to AIDS. You can't live, your immune system cannot function without these helper T cells. So the classification of HIV, according to what we've learned previously about how viruses are classified, it's an animal virus, more specifically a human virus, double-stranded RNA, so there are 2 copies of RNA within the capsid, and it's a retrovirus, meaning that the RNA is going to be reverse transcribed into DNA once it enters the cell. Once the RNA gets into the host cell, it's going to be reverse transcribed into DNA. It's a temperate virus because that DNA, once it's reverse transcribed, is going to incorporate itself into the chromosome of that CD4 helper T cell. And it's going to remain there, and that CD4 helper T cell may even undergo cell division and pass on that pro-virus to its to daughter cells. So you can end up with a whole population of helper T cells that has the pro-virus just waiting inside to someday be expressed and result in the destruction of the cells. That can be quite a while. It can be, in somebody who is not being treated for HIV infection, it can be as long as 10 years before the pro-virus is expressed. If a patient is receiving treatment, it can be indefinite. In other words, the latent period can last for the rest of their life with antiviral drugs that keep the pro-virus genes from being expressed. So as we see here, the full description of infection, the HIV glycoproteins that are on the surface of the membrane envelope bind to CD4 receptors. The viral envelope fuses with the cell plasma membrane, and the viral capsid enters and breaks down. So that's what we're seeing happening here. The viral reverse transcriptase makes a DNA copy of the viral RNA, which incorporates into the host cell DNA as a provirus. After a latent period, the viral DNA is expressed, making copies of the viral RNA and proteins that self-assemble. So here we have RNA, viral RNA, being made from the viral DNA, the proviral DNA. And that RNA is going to, not only copies of that RNA are going to be incorporated into the new viruses, but the RNA, some of that is messenger RNA that codes for viral proteins. So viral proteins are going to be made. And the viral proteins and the viral RNA, copies of the viral RNA, come together and self-assemble. And then they bud from the cell. So new virus particles bud through the plasma membrane, and they surround themselves by a membrane envelope that's derived from the host cell. In other words, as they're budding, they wrap themselves in the host cell plasma membrane. When it comes to viral evolution, they probably appeared on Earth after cells evolved because they are obligate intracellular parasites, OIPs. Obligate intracellular parasites. They have to replicate by getting inside a living cell. So it's, the question, which came first, the chicken or the egg? Well, because viruses can't survive without living cells, then it's speculated because we don't have a time machine to be able to go back and see what happened, that viruses must have evolved or appeared after living cells evolved. They probably developed from the genetic material of their host, first as naked DNA or RNA, just in the environment, DNA or RNA that escaped from inside the cell and then got inside other cells, and then later evolved capsids to better survive and gain entry into their host cells. They don't spontaneously generate. In other words, they don't appear out of nowhere. There's no such thing as a new virus just spontaneously generating from the jungle or anything like that. They evolve from pre-existing viruses, and they may jump from species to species. And an example of how they evolve is the flu. The whole reason that we need a different flu vaccine every year is because the flu virus mutates and evolves and changes every year. And the flu virus is an RNA virus, and that explains why it evolves so quickly. RNA viruses evolve more quickly because RNA is less stable than DNA. So it mutates. It has a higher mutation rate, and that leads to a higher evolution rate. Not all viruses or viral infections cause disease. You are assuredly have benign infections right now. In other words, there are viruses that are infecting your cells that are not causing you to be sick. We're all carrying benign infections all the time. They cause disease by destroying cells and interfering with normal function or development. Antibiotics have no effect because, even the word antibiotic means against living things, right? Bio means living. Well, viruses aren't living, so antibiotics aren't going to be ineffective against viruses because you can't kill something that's not alive. Antibiotics work for bacteria because bacteria are alive. What we do have against viruses is vaccination. So vaccines prevent viral infection by teaching the immune system to recognize the virus before infection. And that's an important note, is that you need to be vaccinated before you get infected with the virus so that your immune system can fight it off. There are antiviral drugs that can slow the replication of viruses in cells and interfere with their replication. But that does not usually completely clear the body of infection. A pandemic is a global epidemic. So, for example, that's why we're looking at this map. HIV is a pandemic, a global epidemic. However, there are antiviral drugs that are being used to extend the latent period indefinitely so that people can live normal, long lives even though they are infected with HIV. One virus or disease that the World Health Organization pretty much wiped off the face of the earth is smallpox. And so this diagram is showing smallpox vaccination. It was just a injection that caused a sore to develop on the skin and left a scar. But they're not doing that anymore. And so the way that they wiped out smallpox on Earth was to vaccinate everyone. The problem is that's extremely difficult. They went all over the Earth and vaccinated every single person they could possibly find everywhere against smallpox, and that's how they wiped it out. Sadly, there are still pockets of smallpox. And the smallpox, the virus is still around in freezers. In Atlanta, there's a freezer that the CDC has. And also there's a freezer in Moscow, I guess, that has smallpox virus, which is kind of dangerous because if somebody could use that as a biological weapon. You, being young, have not been vaccinated against smallpox. So if someone were to break into one of those freezers, steal the smallpox virus and release it into the environment, you could be infected with smallpox. I don't mean to scare you, but it's possible. The bottom line is that vaccinations are extremely important to prevent infection by viruses, and that's really the best weapon that we have against viruses.